Okay, so the plant, will be propagated, by one system called tissue culture without involved fertilization. So the aim of this method is to produce, the progeny consists of similar genetics identity, with their mother plant. Okay, so it can be used to produce numerous plant rapidly without fertilization. Tapi untuk tissue culture, of course, we conduct in the lab lah. Okay, so a few things lah that we have to think about tissue culture is about the cost lah that we use to produce new plant, especially the preparation of the media, the management of hormone use, and then few things lah. Okay, so this is our outline for today. Why we choose vegetative propagation? So what is clones and advantages of clones? And clones also have disadvantages. And then how to induce genetic variation. And the common principle lah. We induce the genetic variation in plant. So when we got the plant with the desired trait, we multiply it, in by using tissue culture process. And then guidelines of selection. Okay. So why vegetative propagation? So plants of a clone are identical genetically. So it is assumed no genetic segregation. But still the variation can occur due to the environmental factor. But in this scope, we focus on the tissue culture process lah. Vegetative propagation required by seedless cultivar. Maksudnya sejenis cultivar yang tidak menghasilkan biji benih lah. So we can do the tissue culture. For example, macam mangosteen, dia ada seed tapi apomatic seed, but it is still can be reproduced by the tissue culture. Ada tissue culture in mangosteen. So as I said lah, the main aim of the production of clone is to produce the plant identical genetically with their mother. So grafting, so why vegetative propagation? Yang ni kita cerita in general lah. Maksudnya vegetative propagation can include grafting, budding, and then stem cutting, leaf cutting, layering, stolon, runner, and so on. So by doing vegetative propagation, we also can combine this some traits. For example, grafting desirable traits in rootstock. So scion combine. So yang bertindak sebagai scion ni adalah plant yang ada good traits. So kemungkinan juga rootstock yang kita gunakan, pokok penanti yang kita gunakan adalah resistant to disease. So can anyone explain or briefly describe to me the factor that affect the success of grafting? Nanti kita ulang kaji sikit. Saya tengok ada tak yang ingat. Boleh tak siapa tolong jawab?
Skill of propagator.
Yalah, skill of propagator. So any propagation method, apa-apa pun skill doktor. Yes, skill lah. Saya skill is acceptable. Other than skill? Other than skill, Nurin. Nurin. The compatibility of the species, okay, between the rootstock and the scion. Okay. So kita kena tengok few aspect macam tu lah. So why vegetative propagation? Another reason is faster maturity. Vegetatively propagated fruits, nut, flower and young. Okay, ni salah satu contoh lah. So dia ambil masa yang sangat cepat untuk matang. Okay. So that's the reason why we use vegetative propagation instead of sexual propagation. Okay. So we can see that, for example, herbaceous perennial require 5 to 10 years from seedling to flowering. 5 to 10 years, ambil masa yang sangat lama. Okay, then untuk pokok orchid, from seed to flowering 6 years. Okay, protocom to flowering 4 years. Untuk tulip and bulb, seed to flowering 3 to 8 years. Okay, protocom to flowering less than 1 year. So kita nampaklah dekat sini the rate of the maturity of plant that propagated by vegetative propagation. So this is the definition of clone. What is clone? Siapa boleh tolong jawab? What is clone? Siapa boleh jawab? What is clone? Janganlah macam tu, cuaca sejuk, semua senyap. Dengan saya menguap. Okay, saya nak Sarah K. Sarah K.
Ya, doktor.
Sarah buat apa? Tidur
Ada.
Dok mana kameranya? Tak nampak muka. Standby bila saya panggil tu maksudnya kena get ready. Be ready and switch on your camera. Izzah pun ngantuk. What is clone? Sarah. Clone is an organism that produce via asexual propagation, either by tissue culture and so on. Okay. Maksudnya without fertilization yang penting. Sekejap. See? What is clone? Clone is the progeny of a single plant which produced by asexual reproduction. Okay, so what is clonal selection? The selection of the most desirable members of a clone for continued vegetative propagation. Theoretically, clones have same genetic information. Yes, clones may have hybrid, and it is maintained through asexual reproduction. So, do you think that any variation occur in clones? Ada tak?
Yes.
Saya pernah muak ni. Okay, Mas, explain why.
Because mutation.
Because apa?
Mutation.
Mutation caused by?
Caused by
Environment, okay, and then induction. okay, by using induced by several chemicals and so on. Okay. So variation can occur in clones due to the mutation and occasional sexual reproduction. So there are several advantages of clones. First, uniformity of population. Okay, maksudnya keseragaman sesuatu populasi klon itu sendiri. Okay, because they have similar genetic characteristic. Okay, so the ability, okay, and the percentage of propagate, okay, are same. Okay, then genetic improvement and selection. We produce the clone, okay, with additional trait in some species, okay, where the some genes, okay, or some character, okay, will be introduced, okay, in the genome of the plant lah, of targeted plant. Sebelum ni dia tak ada karakter tu, tapi nak bagi dia ada karakter tu, so we have injected that gene on that plant. Okay, so when it is stable, okay, it will be reproduced, okay, via propagation, asexual propagation, okay, to produce clone that already improved lah. Okay, and then control of phases of plant development, as well as combine more than one genotype into a single plant. So this all about advantages of clone. Kena ingat ni Okay, instead of advantages, there are disadvantages of clones. Okay. For example, monoculture. Siapa tahu apa dia monoculture? Does anyone know apa dia monoculture? Tahu tak?
Tak.
Monoculture adalah apa yang kita buat masa kita buat dekat penanaman gutta perca itu. Kat situ itu adalah one of the propagation Kita propagate through the seedling. Okay? The cultivation of single crop in a given area. We were provided, okay, by Telecom 300 of gutta perca seedlings. Clear? Okay. So the seedlings, okay, are planted in a given area. So this situation, we call it as a monoculture. Okay. So potential of genetic variation. Okay. Still, clones can induce genetic variation. Okay. Actually, there are two types of genetic variation. First one is genetic variation, which is not reversible. And then, apa dia?
Epigenetic.
Yes, good. Okay, which is, apa dia? Sebaliknya lah. Siapa yang jawab epigenetic tadi?
Srina.
Oh, ni Srina. Okay, okay. Saya bila dengar suara student saya automatically bersemangat. Walaupun hari ni penuh dengan jamuan makan. So saya pun memang ngantuk je lah. Okay. And then, potential for systemic pathogens. Okay. Kalau korang tengok gambar pokok yang belah kanan tu pokok apa?
Kelapa sawit.
Pesawit.
Kelapa sawit. So bawah tu scientific name untuk that pathogen kita panggil apa? Species apa?
Ganoderma.
Terror lah mas. Ganoderma boninens. Dulu, terror. Masa saya kerja MPOB, okay, the main pathogen on oil palm is Ganoderma lah. Kadang-kadang kita tengok, saya based on experience saya lah, kita pergi satu ladang sawit, dan ladang sawit tu dia ada penjarakan sosial. Maksudnya dia ada tanaman selingan. So masa tu saya melawat oil palm plantation dekat Parker. Okay, macam-macam lah kita jumpa. Ular kita jumpa, ulat gonggong kita jumpa. Okay, but saya akan tengok tau, ada beberapa pokok luar dia macam okey tapi dalam dia dah kosong. Kosong tu maksudnya hancur disebabkan this pathogen which is Ganoderma boninens. Okay, and then you can see lah ABC tu maksudnya ada variation, phenotype variation on fruits. Itu semua buah sawit. Buah sawit ada 3 jenis. Okay, and they are classified based on different varieties. Dura, Piscifera and Tenera. Dura kacuk dengan Piscifera, and then you got Tenera. Piscifera act as a maternal plant, manakala Dura as a paternal plant. The commercial one that we use is Tenera. Okay, this Tenera will be multiplied, okay, by researcher from different research agency, okay, to produce rapidly lah. Okay, so these 3 reasons are disadvantages of clone. Monoculture, still potential for genetic variation and potential for systemic pathogen. Ingin saya highlight lagi satu, clone dia ada genetik, theoretically dia ada genetik karakter yang sama kan. So dia punya ketahanan, resisten terhadap penyakit pun sama. So bila satu pokok klon dalam satu kawasan which is attacked by pathogen, so ada kemungkinan satu ladang infected. Okay, so this is disadvantages of clone. Nampak tak kat sini berbanding tadi kalau kita tengok kalau kita tengok advantages lah. Sebab kalau advantages, we can say that the uniformity of population, keseragaman, so we can get banyak anak pokok yang seragam lah. Pada masa yang sama, genetic improvement in the selection, control of phases of plant development, combine more than one genotype into a single plant. Okay, buat transformation first ataupun induce genetic variation. So once the treated plant is stable, so we multiply the plants through tissue culture. Okay, what cause phenotypic variation within clone? Within clone ni maksudnya sepatutnya dia consist of similar genetic characteristic. Tapi kenapa still ada phenotype variation within clone? Still, when we said that phenotypic variation, mesti ada genetic variation because the genetic variation is expressed by phenotypic characteristic. Betul tak? Okay, so we go to the definition of mutation. Mutation is a permanent genetic change involving some part of the DNA molecule. So number 1, okay, the cause of phenotypic variation, which is rearrangement of bases in the DNA structure. Okay, we call it as a point mutation. Number 2, rearrangement of part of the chromosome, such as the deletion of specific sequence, duplication of several sequence, translocation, okay, between the sequence, and inversion. Okay. Number 3, addition of subtraction of individual chromosome. Okay, which is, we call it as a aneuploidy. The chromosome in the organism will increase and will be decreased, okay, due to the variation in the DNA, in the genome. and then multiplication of entire set of chromosome, polyploidy. Okay, usually this thing can be done by treat the targeted plants with chemicals. Kalau kita tengok pokok buah besar, okay, kita buat treatment chemical. Okay, for example, kenapa buah strawberry tu besar, doktor? Mas tanya saya. Because I already treat the genome of strawberry with some chemicals. Okay. So kan, saya letak gambar ni. Korang tengok kan, ni based on my research. Maksudnya apa yang saya dapat, finding saya dalam my PhD research lah. So in my PhD, I studied on mangosteen. Okay. And the variety of mangosteen, which is mustard. Mangosteen biasa, I call it as a common mangosteen with a round shape one. Okay. While its variety, which is famous in Pahang, we call it as a mustard with oblong shape. Bujur, isi dia lebih manis. Pernah makan tak buah mustard? Guys?
Tak.
Tak. Jepun mengecewa ke tak?
Pasti.
Okey, tak apa. Nanti kalau ada, insyaAllah lah saya bagi rasa. Tapi kalau korang jumpa manggis bujur ni, please beli. Okey, dia bukan bujur taman ni. Dia dekat muncung tu. Okey, dekat bawah sepal dia tu. Okey. Sorry, dekat bukan sepal. Saya tak ingat nama dia. Yang bila kita kira kelopak tu, kita boleh estimate berapa ulas buah manggis. Pernah buat tak? Tahu tak pasal ni? Kan dekat muncung dia tu, ada bunga. So, korang boleh tengok. And then, instead of korang tengok berapa ulas, korang tengok kalau yang tebal tu, kan dalam antara bentuk bunga tu, kalau ada yang tebal sikit tu, dia ada seed. Tu cara tengok Based on my study, saya nak ceritanya, mangosteen ni, dia ada a nucleotization in its genome. So ada pertambahan dan kekurangan kromosom supported by chromosome data in my study. So I found that, okay, the chromosome number of mustard is higher is compared to the common mangosteen. Okay, this is the reason. The occurrence of phenotypic variation when the complex genome in mangosteen trigger a nucleotization. So, it will be expressed, okay, phenotypic lah, secara phenotypic. Okay. So itu apa yang saya punya study lah. Kalau kita tengok macam alah ni based on teori saja, tapi I try to say is ini adalah based on my research lah. Okay. So instead of kita sebut tadi mutation, the arrangement of the DNA, okay, and then nucleotization, polyploidy. So faktor lain that cause phenotypic variation, tadi tu kita sebut genetic variation lah. Environment by genotype interaction. Okay, kat sini yang kita belajar dalam plant physiology. Okay, drought stress, high salinity and so on. It can kalau kita tengok, kita ada banyak fasa of study tau, genome. So after genome, kita akan pergi kepada transcriptome. Transcriptomic ni focus on RNA. So genome DNA. So after transcriptomic, kita akan go to the proteomics, which is the study of protein interaction. And then barulah the character of the species. Nampak tak? Dulu yang kita belajar molecular biology tu, kalau dalam genetik pun korang belajar kot, transcription, translation tu, ini adalah dia punya application lah, which is cause variation and so on. Okay. Sebab dia akan ada reaksi ataupun reaction when the environment punya stress lah. So that akan trigger the genetic or genome to be altered lah. Okay. And then setelah itu permanent genetic variation. Okay. Of course permanent genetic variation ni macam tadi lah. Okay, yang saya cakap tadi. mutation, okay, translocation of the DNA sequence and so on. And then infection by systemic pathogens. So it will also cause phenotypic variation. So genetic variation induced by mutation. Actually, there are several mutagenic agents. Okay, ada tak yang berminat study pasal ni? Hello, kelas tidur ke?
Tak ada.
Saya tengok muka seseorang ni. Takut saya je cakap seorang ni kan. Rina mana Rina? Rina tidur gila. Tak menarik ke tajuk saya ni ke semua ngantuk sebab petang? Macam tu petang. Rina pernah study tak pasal this topic? Genetic variation induced by mutation. Pernah tak? Dalam kelas genetik ke? Macam pernah? Okey, betul Kalau tidak saya tanya Dr. Latif. Ni saya call Dr. Latif sekejap. Tak ada. Okay. Actually, kalau korang tengok kan dekat Cameron ke, ada certain plant yang dia induce untuk besar. And one of my research interest adalah mutational breeding. So this part, bila kita induce mutation ni adalah under mutational breeding. One of my master student working on orchid. So in her research, dia daripada UPM lah. Tapi I'm one of the co-supervisor lah. So in her research, he want to create another variety of orchid which is bigger than normal size. So what she did is to treat the orchid with selected mutagenic agents. Such as, of course, chemicals, hydroxyurea and hydroxyquinoline. And some are induced from tissue culture product. So alhamdulillah, she finally berjaya lah untuk develop orchid yang lebih besar sikit. Sebab orchid yang lebih besar ni currently menjadi permintaan industri tau. As a ornamental plant. Walaupun tak jadi semua tapi ada certain yang jadilah. So as I said lah, mutational breeding, developments of new clones with the use of mutagenic agents. But this thing dia masih lagi tak dikira GMO, genetic modification organism lah. Because we did not introduce new gene to the plants. So another methods for mutational breeding, instead of chemicals, we also use gamma ray and neutrons. So gamma ray ni kita akan pancarkan lah dekat that plant yang kita mahukan mahu induce genetic variation. So gambar bawah ni we can see that different ploidy level of strawberry. Strawberry yang besar tu of course it consists of larger genome as compared to the right one yang kecil tu. However, it can be produced through mutational or mutagenic agents. Clear semua?
Yeah.
Betul.
Ni ada part-part genetik lah. Sebab kalau saya cerita pasal clonal selection ni macam I think quite boring jugalah. So the treatment can be done by attack specific part or specific branch. For example, kalau kau tengok gambar kiri tu, satu cabang pokok dia still buah dia size yang sama, normal, 2N kecil. And another pokok kita inject dengan colchicine untuk this branch. Lah, saya nak tanya soalan IQ. Rasa-rasa kalau dua-dua branch kita buat cutting, agak-agak buah dia akan sama saiz ataupun yang kanan akan maintain lagi besar daripada yang kiri? Siapa boleh jawab soalan ni? Kita application ni of the of the mutational breeding, but we use selected branch by injecting the chemicals on the selected branch. Then after a few months we can see that right branch will produce larger fruit as compared to the left branch. So kalau kita nak buah yang besar tu, kita boleh if we want to propagate yang buah lagi besar tu through cutting, so kita nak ambil branch yang lagi besar tu ke buah lagi besar ataupun branch yang mana-mana boleh? Siapa boleh jawab soalan ni?
Branch lagi besar lah.
Okay, sebab kita tak treat keseluruhan pokok dengan mutagenic agent because kita selected by branch. Melainkan kalau daripada seed kita dah treat pokok tu dalam mutagenic agent. Clear? So cutting of the dah ditulis lah, cutting of the diploid branch will produce diploid plants with diploid flowers, fruit and seed. We can select lah branch mana yang kita nak jadikan dia tetraploid. Tetraploid is referring to the 4 set of genome. Okay. So, pernah makan tak seedless watermelon? Siapa pernah makan?
Tak pernah.
Pernah? Seedless anggur?
Yang
Pernah kan? So, tahu tak proses macam mana nak menghasilkan seedless ni? The character of seedless is contributed when the genome of the organism is triploid. Maksudnya if dia ada 3 kromosom, so the character adalah dia apa dia? Dia tak produce seed. Dia seedless. Okay, so if you can see from the diagram, first kalau kita nak develop triploid 3 set watermelon, so we have to fertilize 2 diploid tembikai. So dia akan jadi 4N. Okay, 4N. So daripada 4N tu macam mana kita nak growkan dia jadi 3N? Ingat tak benda ni?
kita dengan 2N lah.
2N campur 2N dia akan jadi 4N. So macam mana daripada 4N tu nak jadikan dia 2N. Okay lah. Yang ini saya bagi jadi homework rumah. Fikir and nanti minggu depan explain how to produce seedless watermelon. Okay? Tak payah buat slide pun, just saya saja yang disembang. Okay? Clear ke?
Clear doktor?
Ya.
Semangat ni.
Ada ini.
Ngantuk ke semua? Ke jamuan raya lagi?
Tak ada
Tak.
jamuan.
Tak ada jamuan. Okay lah, selingan lah 5 minit, takut korang ngantuk kan. Field trip report, video dah siap lah. Tak sabar nak tengok ni.
Belum.
Belum. Oh, bila siapnya? Esok?
Weekend.
Weekend. Oh, weekend barulah apa nak ajak Mas makan-makan. Cik, Mas je kan? Tak ada gurau-gurau.
Boleh doktor, boleh.
Okay. So, okay, saya saja je nak menghilangkan rasa gundah gulana dan mengantuk. So far bab ni clear ke? Dia just cerita distinct lah. Sebab nanti my plan lah macam saya cakap tadi, at the end of the lecture, mungkin last class lecture tu saya just nak simplify all the tools that we learn during class. Pasal tools sahaja. Okay? So kita fokus on propagation tools. Yang ni kalau korang nak baca this part, which is okay. Okay, this is part from the course outline lah. Okay? Tak ada masalah. Okay. Then guidelines for selection. Tadi kita dah cerita pasal clone semualah. Okay. So of course guideline for selection. Selection daripada stok lah. Maksudnya stok yang kita nak apa? Nak pilih untuk jadikan dia clone. Okay. Of course, we have to know the history of production of the stock. Banyak ke tidak ataupun macam mana. Okay. Plus that have been inspected. Maksudnya free from the pathogen and so on lah. Okay. If genetic disorders are common for the stock, conduct a vegetative progeny test. Okay. Conduct frequent visual inspection. Conduct test planting and inspect for trueness to type. Tapi yang conduct test planting and inspect for trueness to type ni pernah belajar ke dalam kelas Dr. Poci? Hello?
Tak pasti.
Tak pasti Okay, in Dr. Poci class, propagation method apa yang dah belajar?
Cutting. Baliklah. Cutting je.
Cutting je?
Grafting, budding.
Cutting, budding, adalah few things lagi kan, karakter yang korang dah belajar kan. Cutting, budding, marketing part saya. Layering sekali lah, and specialized structure pun daripada saya. Budding yang dia cover tak budding?
Rasanya ada.
Rasanya ada lah, tapi mungkin budding dia couple dengan grafting lah. Sebab method dia sama, cuma budding dia guna mata tunas lah. Okay. Okay, so guidelines for selection. Tadi pun for selection lah. So this is a few guidelines lah of the selection before we do the cloning punya part. Okay, visual inspection and phenotypic selection. Source selection based on phenotypic appearance of the source plant. So we have to observe, okay, the phenotypic appearance of the source plant, whether it is okay or not before we do the selection for cloning, okay. And then the second one, which is genotypic selection. Source selection based on phenotypic appearance of the vegetative progeny. And then vegetative progeny test, which is vegetatively propagating progeny to test their ability to reproduce the source plant. So all these guidelines for the selection, which is from the stock which is referring the original of the plant lah. Okay. So this is several methods that used in order to detect the variation that occur in the plants, okay, after and before clonal propagation. Okay. So pedigree production programs. First, identify individual plants with the clone that are genetically true and free from previous pathogen, of course. And then step 2, maintain source plants in a protected foundation block located at to prevent reinfection. And then multiply source and distribute to the public. Maksudnya kita pilih, kita start daripada how we select the stock plant. And then what are the characteristic, of course, phenotypic punya character that we have to observe before do multiplication. Then maintain source plant in a protect foundation block located to prevent the infection. And then number 3, multiply source and distribute to the public. Everything we do, of course, the contribution to the public. Okay? Clear semua. So I think today's class already complete. Okay, principle and practices of clonal selection. So in this topic, you have to know the definition of clone, the disadvantage of clone, okay, and then the definition of the clonal selection, and a little bit about the variation, because we want to learn about selection. Okay, the guideline for the selection. Of course, bila kita sebut selection, we have to observe the phenotypic and genetic characteristic of the stock plant. Okay, so we also should know the tools that can be used to determine the variation that occur in the clonal plant. For example, macam ni lah, PCR amplification of the DNA. Mungkin boleh guna ISSR, okay? And then fingerprint dendrogram lah, which is yang kita buat, kita sequence, certain sequence, kita sequence daripada primer, certain kawasan tu, and then kita amplify, dan kita create the dendrogram tree based on the sequence data. So we can observe the variation on that data lah. And then pedigree production program, the step 1, step 2 and step 3 lah. Okay. And then the last one. Okay guys, clear ke? Simple saja kelas ni hari ni. And then I think the next week kita belajar specialized structure, and then followed by your presentation. And before presentation, okay, kita ada kelas ulang kaji, revision class. Okay ke macam tu kalau saya buat? Sebab 2 chapter last ni which is yang dah tak touch propagation. Tapi untuk yang revision class tu, saya wajibkan semua ada, sebab yang itu memang kita akan fokus 100% lah on the propagation method, which is nanti kita akan recall method daripada Dr. Pochi sampai saya, and then we know the difference among each techniques and its application. Sebab soalan saya tak direct. Terus terang saya cakap. Tak macam soalan quiz. Soalan quiz is easy, tapi kalau untuk propagation, seolah-olah soalan saya itu macam korang yang menanam pokok tu. Korang yang initiate the idea how to propagate the plant, whether it is suitable on this plant. For example, this technique suitable tak untuk plant ni? This technique suitable tak untuk plant ni? So ada satu soalan tu kalau salah atas, bawah semua salah. So be careful. So kalau tak faham tanya, itulah kegunaannya revision class saya. Untuk kelas yang ke-13. Kelas yang ke-14 adalah presentation daripada you guys lah. Hari Selasa tu. Okay. Jumaat ni esok ada topik yang memang minor lah. Sekejap je. Sejam macam tu lah. Which is yang saya nak sentuh specialized structure, stem semua tu. Saya nak perincikan lagi lah. Okay. And then minggu depan Selasa pagi revision class. And then hari petang tu Selasa presentation. Okay, macam tu jadualnya. So dah nak habis dah pun kelas. Propagation method pun dah ni. Tapi itulah saya risau bila saya tanya propagation teknik ni semua macam ingat tak ingat je. Semua senyum je kalau saya tanya tu kan. Saya pun tak tahulah. Semua meriah je.
Doktor nak tanya.
Siapa
Sarahani.
Sarahani. Oh, cucu doktor. Cucu Cikgu Rosia. Saya dah ingat lah. Okay. Yeah.
Yang
Okay.
tadi doktor kata monoculture tu disadvantage bad thing lah.
The disadvantages of clone. Okay, satu kita kata monoculture. Monoculture tu yes, dia satu pokok di dalam satu kawasan yang sama. And then daripada monoculture tu, the resistance of the plant to the disease. Sebab bila dia ada genetik yang sama, so dia punya resistance tu pun lebih kurang sama. So when one plant is infected by ada patogen, so semua nanti akan kena sekali. So kalau kat ladang sawit tu, this is a big problem lah. Kalau satu pokok dah kena ganoderma, so memang pokok-pokok lain tu walaupun di luarnya kelihatan baik, tapi kalau belah batang bawah pokok tu memang sebenarnya tak okey. Based on my experience lah. And then kalau macam kat ladang sawit tu ada yang dia buat tanaman selingan. Tapi kena tengok. Tanaman selingan tu dia korang perasan tak bila korang tanam gutta perca tu, kawasan siapa yang buat dengan saya kawasan atas tu? Mas dengan Nadia kan. yang buat dengan saya kan dekat atas tu. Pasal tak jarak dia beza dengan jarak pokok yang getah paca yang belah sana, hujung dekat depan sana, dekat hutan sana tu.
Perasan.
Sebab dekat kawasan plot yang saya mas dengan Nadia punya tu, Nurin, adalah untuk buat tanaman selingan. So kita orang ikut SOP sawit. Berapa space? Ini pun korang kena tahu. Mungkin tak ada dalam lecture note tapi mungkin ada dalam soalan exam. Berapa space yang kena ada daripada satu pokok ke satu pokok yang lain. Itu saya consider as a monoculture untuk permulaannya. Sebab kita diberi anak pokok getah pecah untuk ditanam di satu kawasan. Tanaman selingan tu kemudian. Tapi getah pecah tu kita tanam from seed. So the life cycle dia mungkin lebih lama instead of tissue culture lah. Tapi resistant terhadap penyakit tu insyaAllah lah okey. Tapi pada masa awal ni kena selalu siram lah. Dan siapa yang volunteer nak siram, bolehlah pergi turun siram. So far kan Lalang tu saya nak tanya kan, untuk batch korang 201 ni, subjek apa yang paling susah untuk body system? Sebab saya tengok survey tadi tau. So ada yang tak isi. Subjek apa yang susah korang rasa?
Statistik.
Statistik.
Statistik Dr. Siti Marponga. Tapi itulah kalau saya nak buat body system, itu di luar department kita. Tapi tak apalah, nanti saya discuss dengan Dr. Zarina. Sebab I'm quite worried, especially based on on your performance lah during final. Tapi kalau midterm propagation itu senang tak? Propagation okey
Okey.
Okey. So berdoalah untuk final sebab final saya yang buat. So memang macam saya cakap kadang tak ada dalam lecture note tapi apa yang kita sembang dekat Mardi. Mardi kan ada benda kecil-kecil yang technically pun kita learn daripada Mardi. So saya pun learn sekali sebab saya pun dah lama tak buat. Okay. So please ingat step. Selain ingat step tu kena tahu the principle of the propagation technique kita guna. Kenapa kita buat ni, kenapa kita buat tu and so on. Soalan dia tak direct. Okay, so macam tu lah. Okay, Sarah ni orang Bangi, so saya menjawab soalan.
Menjawab alhamdulillah.
So I think okay lah, minta maaf sebab saya memang menyelesaikan kelas lah. So that nanti korang ada 2-3 minggu extra yang korang boleh fokus study genetics and so on. Sebab saya tahu assignment semua saya dah bagi, and then quiz saya dah bagi tadi, field trip pun kita dah buat, lab pun saya dah bagi. Saya tunggu submission daripada korang je. Okay. So by 5th June, insyaAllah lah carry mark dah ada. Okay. Midterm tu kena tunggu Dr. Poci lah. InsyaAllah. Okay, so kat situ bila dapat carry mark tu korang dah boleh target dah. Korang macam mana korang nak dapat A untuk di subjek. Tapi basically plant propagation insyaAllah semua okey-okey saja. Sebab basic subject for plant science student. Okay. Semua orang menanam and even dia tak ambil plant science pun dia propagate pokok. Nanti kita tengok pakcik dekat Jabatan Pertanian kan. Saya tak nak bawa korang kan. Dekat Bukit Goh. Makcik tu dia buat grafting relax gila. Tak macam kita lah kan. Tak macam saya lah. Korang pun dah terror lah sekarang. Dia dah darah tangan. Buat marketing kena duri pokok limau. Macam Izzah dah terror lah buat buat grafting yang itu, belajar dengan Cik Sabri kan. So I hope ni lah, tapi enjoy lah field trip hari tu. So saya harap bukan sekadar enjoy tapi korang dapat lah banyak benda. Cakap ni memang skema lah, tapi bila korang nanti dah berada di umur-umur sebaya saya, mungkin korang akan berfikir dengan lebih dalam lah. Sekarang ni enjoy dulu kan. Okay, so quiz dah ada, quiz dah jawab alhamdulillah. So hari ni kelas kita pun dah settle. So Jumaat kita jumpa 8 sampai 9, sekejap je kelas. Tak lama. And then hari Isnin lah. Hari Selasa pagi revision kelas tu kena datang, kena join. Kalau tak join ada masalah tak apalah, tapi kalau tak apa-apa untuk final T, ni lah. And then petang presentation. Okay. So I think that's all lah for today. So thank you semua. So tadi pun saya tak buka kelas. So I think kita sampai sini je lah. Ada question ke pasal assignment ke? So far saya tak ada nego yang baru, tapi nanti saya akan table untuk deadline yang baru untuk assignment tu. Yang itu siapa? Izzah minta. Okay guys, so kalau tak ada apa-apa, kita stop sampai sini saja. Thank you for joining today's class. So I hope that semua info dapat lah. Revision class kena ada minggu depan, hari Selasa. Okay. So, okay semua guys. Bye. Assalamualaikum.
Salam.
Take care semua.
Thank you, Doktor.
Waalaikumsalam.
Doktor.
Okay.